Hi, my name is Giulia. I'm a second-year PhD student at University of Teramo and Istituto Zooprofilattico Sperimentale of Abruzzo e Molise in Italy. My project aims to develop internal protocols for environmental DNA analysis in marine ecosystems. But what's environmental DNA, and in particular marine eDNA? Let's think about the ocean as a glass of water. If we filter the water and extract the total DNA, if we look at it closely, we're gonna find different traces of DNA from fish, algae, plants, viruses and bacteria and other organisms, humans included. Following a metabarcoding approach, we amplified the mitochondrial 12S gene specific to fish in order to identify every species in our glass. While this technique has been highly used for monitoring biodiversity profiles and searching for target species, such as protected or invasive species, there is a need for a correct interpretation of the data. In the first phase of this project, we tested our protocols using mock communities. We started with a free species mock community, we sequenced them and then checked for false negatives and positives. We created more complex communities with 13 species. We sequenced them and then adjusted the parameters. And finally, we tested the improved protocols in more real but still controlled environments. In collaboration with the Marine Institute of Ireland, we took samples of water from a salmon fish farm at different distance from the fish cages in order to delve deeper into the fate of DNA and understand how far it can travel from the source. We then tested our protocols in a wider environment along the southwest coast of Ireland. In collaboration with the Irish Whale and Dolphin Group, we jumped on board the research vessel Celtic Mist during a dolphin monitoring leg. We took samples of water during every sightings to get information on the co-presence and interspecies interactions between marine mammals and fishes. We both use active and passive methods for sampling marine eDNA. Filters were then stored before starting with the DNA extraction process, once back in the laboratory. Different extraction kits were compared, evaluating the efficiency in terms of DNA concentration and the quality of the DNA extracted. DNA libraries were prepared following a two-step approach. The libraries were then sequenced using Illuminal technologies. And finally, the bioinformatic analysis was carried out in order to understand which species of fish was present in the samples.